Hello students, a warm welcome to all of you in Idealize Academy. In today's lecture, we will discuss about the last portion of vaccine designing. In last lecture, we will discuss about the vaccine construction. In today's lecture, we will discuss about the docking. For docking, we use Clustro. Whenever in Google, we simply search for Clustro. In Google, you simply search Clustro. Then open this first one. The software is used for the protein-protein interactions. Then click on docking. Dock. Yes, give the job title, and you also upload your receptor and ligand. This ligand, that is the model number 1, that is generated by the T-Rostra. Okay, then simply click here and dock. Okay, now your your processing will start. Okay, then after some time you will get your result. Okay, the software automatically generates of models, but I use 10. For downloading and display model, simply click here for download, and this first one is more appropriate, that's why I select this one. First downloading this one, is simply click here. Whenever you click here, then this model is downloaded in your PC. Okay, now this is the publication. Okay, because I downloaded this first one. Why? Because this first one is more appropriate. Okay. Then for my molecular simulation in Google, I simply search iMod. Then open this home page of iMod. Then browse my PC, and my model one is this one. And this is the protein and ligand interaction. So I open it for the result. Wait, now this is the result of iMod or molecular simulation. Okay, this is the result. Okay, this is the result, and the principle and the algorithm behind this result is NMM mobility. Okay, this is for the deformability, chain, SPK format, hydro, beta factors, aerofield, Arvin. Okay, this is for the spin. This is the deformability and atom index. Okay, and this peak point denote this hinge denotes the high deformability. This is a beta. Now this is the beta factor graph. This is a beta factor graph. This is beta factor graph is taken from the corresponding PDB field. Okay, and calculated from the NMA. It is obtained by the multiple NMA mobility. Okay, we are aware that many PDB files average NMA model contain no beta factor. Actually, beta factor is column gives the average RMS value. Okay. Now, this is the eigenvalue graph. This eigenvalue graph is normal mode represent the motion stiffness. Okay, its value directly related to the energy required to deform. Lower the eigenvalue, more the appropriate result. Variance, this variance is inversely proportional to the eigenvalue. Okay, and this is the covariance graph. This covariance graph is between the pairing the residues. Okay, and this red color denote the experience correlations, white portion denoted the uncorrelated, and blue is for the anti. Okay, and this is the stiffness graph. More this gray region means more the stiffness. Okay, this is the iMod result. Okay, now for the amplification for the cloning process, we use a database and result of cloning. And the docking analysis and molecular simulation step. Now we understand the vaccine expression analysis. Okay, for this we use transback inverse. We use this transback inverse to convert the nucleotide amino acid sequences into the nucleotide sequences. And this is our this is the amino acid sequences of this vaccine. Now I optimize this amino acid sequence. For optimization, we use a software GCAT. Codon optimization. For this, I simply open this one. The sequences and paste. Okay. Then we select the organism in case of Homo sapiens. Then select the enzyme histidine. Select what the cleavage size with this restriction enzyme. Okay. Then now these are the optimized sequences. Okay. Now I copy the sequences and paste the sequences. Okay. For cloning and expression analysis, we use a software like AnyBridge Golden Gate. Then I open this one. Okay, this is the home page of this software. Okay, then we click and quick start. This is the overview of this software. Then we click on add sequences. Okay, then paste the sequences and copy this improved sequences and paste. Okay, processed, this one, change, lane and add. Now this is your final result. This is the ligation, fragment ligated. Okay, this is the fragment ligated and this is the complete information and these are the primer. This is the forward primer for the sequences and this is the reverse primer. Okay, by the help of the software we easily automate your ligand into the vector. Okay, for individually downloading the vector we use a software like Adzine. Okay, and open this one. This is the database of the plasmid and I simply search my plasmid like PVA, like PVR and search. Okay, let's suppose this is the overview. For enlarge, click this one. These are the amino acids. This is the complete information about this plasmid and restriction enzyme. Simply click on view all the sequence. Now these are the sequence of this amino acid. This is the sequence of this plasmid. Okay, for downloading the GenBank, click here. For SnapGen, click here. For analysis, click here. Okay. This is the map. This is the sequence. This is the complete sequences. And this is the enzyme. This is the list of enzyme, their restriction site, the number of cutting position, features, primer, blast, and analyze that. Okay. Okay. For the cloning process, we use another software, that is a Benchling. For Benchling, I simply open this Benchling. Okay. Then open this software. Then sign up. Then enter email ID and sign in. Okay, wait. Okay, then this interface will open. Okay, by clicking here and DNA and amino acid sequences, simply upload your sequences. Click here, then DNA and amino, click DNA and amino acid sequences. Okay, and copy these sequences and paste, then create. Okay, now this is your sequence. These are the restriction enzyme. Okay, and this is the exact position. Okay, for the primer, let's suppose I want to use this plasmid. Okay, so go to the sequences, back, this is the sequences, then copy the sequences, bench link, new sequences, plus bench link, new sequences, paste and create. Now this is the plasmid. Okay, in this plasmid there are number of options are available. Here this is your sequence and this is your plasmid. Okay, and these are the restrictions. Okay, simply cut and assemble your sequences. Okay, but this software is automatic software and very easy software. Okay, and by the help of this software you also create annotation, primer, translation, new amino acid sequence, DNA as well as, and we analyze in analysis, run primer 3, submit in NCBI for the blast, analyze the translation and optimize the Gaussian codon like forward and reverse. Okay, for the cutting process you simply digest, for the cloning digest, then choose the enzyme like FSP EI1, select this one, then run digest, this one and run digest. Now your enzyme will cut. Same for the sequences, is cutting F S, this one, select and digest. Okay, for cutting use this for assemble, click on this assembly. Whenever you click on assembly, then your sequence is assembled in your plasmid. And this is all about this vaccine expression and analysis, all about the 2-day sessions. Follow ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn. Please like, share and subscribe ITLS Academy. Thank you.